Good morning, everybody. We're just waiting for everyone to join here. People are trickling in to the webinar. We'll get started in a couple minutes. Okay, good morning, everybody. It's 8:03, so we're going to go ahead and get started. Welcome to the, I think, 5th installment of the Research Coordination Webinar for junior researchers, defined as the hardworking assistant professors, postdocs, and PhD students who have been doing their service to the pandemic for the last 9 months. This forum is a place for everyone to share information, hear more than anecdotes from people about their research, and be able to ask questions and learn from each other. Today, we're really excited to have 2 people. We have a PhD student from Rice and a postdoc who works at Nevada Water. And our first speaker is going to be Prashant Kalvapalle, I think I said his name correctly, who's going to talk to us about their experience in Houston. And before we start, I just want to explain how this is going to work. So we're not monitoring the chat. What we are doing is monitoring the Q&A. So if you have a question during the webinar, please type it into the Q&A. And then there's the, you can upvote questions if you like the question and you want it asked. And moderating the questions will be Kyle Bibby for the first speaker for Prashant. And then for Katerina, Katerina, Rolf is going to do the Q&A for that. So the speakers will speak for 20 minutes and then we'll have some Q&A. So with that, I'm going to pass it off to Prashant. Thank you so much for joining us today.
Hey, good morning, everybody. Can you hear me?
Yes.
Yeah, so my name is Prashant. I'm a graduate student at Rice University, and we've been working extensively for the last 6 months in monitoring the wastewater in Houston. So I'm going to talk about our efforts in 2 things. One is developing methods for detecting the SARS CoV2. And the second one is how do we use the data to take some actionable action, and then predicting the long-term clinical positivity rate using the wastewater data. Because it's early in the morning, it's a good idea to remind everyone, why are we detecting this virus in wastewater? First thing, we are able to use wastewater to detect the signal of the virus from many people at the same time. So it's a community scale and it's very cheap compared to that scale. And we are able to do this rapidly, and we can get samples once every week like we have been doing. And we use this time series data in a very simple manner compared to like testing individuals once every couple of weeks or something. So because it's a very nice thing to do, the challenges to do the wastewater sampling here is that the SARS CoV2 virus is very dilute in wastewater compared to a human clinical sample. And it also has so many inhibitors. So the methods that we take to extract the virus and the RNA from the virus actually impact a lot on what exactly we can detect in the wastewater. And since this is a very rapidly moving field, because of the pandemic, everyone has been squabbling to work on this. And there's so many methods that you find in literature, it's hard to find which is the right methods to choose. So what we decided to do is, right when we started this epidemiology effort, we simultaneously were developing or trying to find the best method and optimize it for our lab's conditions. So we were having many considerations such as the throughput. We wanted to process 50 samples every week. And then we also were taking into account the sensitivity, like we wanted to detect low amounts of virus and the cost of developing these methods and starting up and everything. So for a start, we decided to compare these 5 methods. 2 of them, A and B, are based on using these electronegative filters. We pass the wastewater through the filter and the virus binds to the filter and the water is discarded. So we have concentrated virus on this filter, and then we elute the virus from the filter, either passing through another solution or by using beads and physically disrupting the filter to extract the virus out. And the third thing we tried was directly putting the wastewater into an RNA extraction procedure. So we call this direct extraction. And the fourth one is to use a filter that has a much tinier pore size than these electronegative filters. So it's a size exclusion based capture of the viruses, and then you throw away the water and anything that's smaller than the virus. And the last one is to use ionic solvent like polyethylene glycol. We abbreviate it as PEG, to selectively precipitate the virus or any proteins and things like that that need a certain water and conditions around them to stay soluble. So if you exclude the water from that using PEG, then the virus is precipitated out. And since it's in a precipitate, you can centrifuge it down and you will collect it in the bottom and you can throw away the water. And so we put the wastewater through all of these methods, and we do the RNA extraction using a machine called Maxwell from Promega, and it's using magnetic beads to capture the RNA. And we are using droplet digital PCR to quantify the virus. So for those of you who are not well versed with the droplet PCR, the way it works is, you can imagine the normal qPCR curve where the signal increases with the number of cycles that you're doing. So this reaction is happening not just once in the whole well, but the sample is partitioned into tiny single droplets, and within each droplet a qPCR reaction is happening. And what you're reading is the endpoint fluorescence. So there are many droplets that have this black cluster. There are many droplets which do not have the template that we're looking for, so that's why they have a low fluorescence. And there's very few that have a high fluorescence, which we are assuming they have 1 or more targets. So when your sample is very dilute, then we are detecting a very rare template like SARS CoV2. We expect that the number of droplets that we count here in the whole sample is proportional, very close to the number of template molecules that was in your reaction. And we use this limit of quantification of 3 droplets per 10,000 that we detect. So we use this from the statistical rule of 3 for subsampling, because we have a larger volume of RNA, but we sample a few of it and then we put it into discrete droplets. So whenever any discrete distributions are involved, you get this rule of 3. And for limit of detection, which I have not talked about and I have not shown in the graphs, that would be, we could define it as detecting even a single positive droplet, but you can imagine if there's any like cross contamination, we can have 1 or 2 droplets that show up in your reaction. So you can also define it based on a blank sample that you run. So you can define it as 1 standard deviation above the blank samples. So now we have done the droplet PCR on all of these 5 different methods. And we notice that this black line here is showing the limit of quantification as I've defined here. Most of the methods, the data points are falling below this limit of quantification. So they have less than 3 droplets per 10,000. So if you process more droplets, let us say, we are assuming that the limit of droplets is slightly more. So the method that stands out here is the electronegative filtration, which we are defining as abbreviating as HA filtration, and bead beating to remove the virus from the filtration. So these 2 methods together are giving us very good extraction compared to all the other methods that we've seen. So we take this data that we are defining it as copies per microliter in the RNA, and because each of these methods have a different amount of wastewater that goes in and different amount of RNA that comes out, we are defining what we calculate as the concentration factor. So for the HA filtration bead beating, effectively 25 microliters of 25 ml of wastewater goes in and 100 microliter of RNA comes out. So if you divide those two, you get a 250-fold concentration. Similarly, we have defined it for all the other methods. So it would be more comparable because we are processing the exact same sample through 5 different methods. It would be more comparable to see the copies per liter of the wastewater, which we are getting by dividing the copies per microliter RNA with this concentration factor. So here it looks like the story is different. There's other methods like direct extraction that have a higher effective copies per liter, but since it's very close to the limit of detection and it's actually below our limit of quantification, even from this graph, we should conclude that the HA filtration plus bead beating is the best method because it's well above the limit of quantification. So not just presence absence, we are using this data for quantitative data trends. One more thing we did is while we were detecting SARS CoV2 in the wastewater, we also added another virus. So this is a bovine coronavirus. We assume it has similar characteristics as SARS CoV2. And we spike a known concentration of this and we extract the same virus, and we are calculating the fraction of what we put in versus what we get out. And here it seems like this trend kind of matches the N1 data that we see. So direct extraction has a higher fraction of recovery. There's some loss in the filtration and bead beating because maybe we are not able to capture all the virus from the filter. So it's probably stuck to the filter. And all the other methods also have losses. So this is 100% recovery, which is 10 to the 1. It's not shown here. So all of our methods are recovering below 10% of the virus that we put in. Another virus that we also detected is this pepper mild mottle virus. It's a fecal marker present in wastewater in high abundance. So you can see this axis is much higher than the SARS-CoV-2 detection axis we have. And so we also added this in because we assume that this is present at a high enough abundance and it's more or less constant across all the treatment plants. So the x-axis in our graph here is showing 6 different treatment plants along with a DI water control, which doesn't have SARS-CoV-2, but it has the bovine coronavirus spiked in. So we took both the data sets we had, one for the surrogate virus and one for the SARS-CoV-2, and we divided one by the other just to enable us to compare across methods. And if we wanted to benchmark our work against existing methods that we took from literature, so we can use this divided normalized fraction of SARS-CoV-2 to compare across all the methods. And it's very interesting to see that previously the variation was along 3 orders of magnitude. It has come down to 1 order of magnitude. So it's most of the data lie between 10 to the 6 and 10 to the 7. So we should conclude from this that our surrogate virus, the B curve, is maybe not identical to SARS-CoV-2, but it's a good enough surrogate to use when you're trying to compare across methods. And the second part of the talk is, once we have the data, what do we do with it? So we have been simultaneously, while developing the methods, we have been working on processing every week 39 different wastewater treatment plants all over Houston. So this map here is showing the geographical area served by each treatment plant. So Houston is a very unique city within wastewater treatment parlance. So there's one very big treatment plant, the 69th Street, which is similar to what other cities have, which is very big centralized plants. But there are many other smaller wastewater treatment plants. And so precisely for 39 total, one of them is very big, and there are few that are really small. So this gives us a very nice picture of varying sizes of treatment plants, and because they are located geographically in different areas, once we get the data, if we plot it on a map here, we are able to see hotspots. So there's like redder spots for a particular week, and then they go away the next week and some other spot shows up. So we can use this data to identify which areas need more health interventions. And we have our amazing Houston Health Department who is sponsoring this project as well. And they have been using our data very effectively by deploying targeted testing and intervention in the particular hotspots that we identify. So it's very heartening to see the data being used real time. And apart from the spatial variation, we also have the temporal data. So because we are sampling once every week, we can do a short-term trend. So last week to this week, we can see which areas increased, and we use that to identify the hotspots as well. But the second thing we do is also a long-term prediction. So all the data we have till now, we put it on a graph, and then we try to relate it to the clinical positivity rate data. Just an overview, because I've talked about many methods. While we were developing those methods, we were also doing this. So we were doing this in 2 different labs simultaneously, just to see how robust our conclusions are when we use different methods. So in our laboratory at RICE, we have developed this protocol for doing the HA filtration bead beating, which I said was the best method from the previous slide. And then we were doing the Maxwell extraction and droplet digital PCR. But simultaneously, the same samples in another laboratory in Baylor College of Medicine, they were doing a slightly different version. So using elution after the filtration, and then using a different company's extraction kit, also using magnetic beams, and they were doing RT-qPCR. So it's very interesting to see that the data from these 2 very different methods are able to map onto the same graph, and they are able to give us same conclusions, because we are doing so we have a statistical team working with us from Rice University, and they developed extensive statistical methods like this method I'm going to describe here. So we have, for the short term, we have data from last week, we have data from this week. And because we have replicates from each lab, and there's SARS CoV2 N1 and N2, both of which we are detecting, 2 different targets from the same virus. We have so many different kinds of data. So how do we make sense of it to see whether it's just random noise, or is there a trend significantly going from one week to the next? So we are using something called multi-level regression model. So it's I've drawn this schematic here. So we have multiple wastewater treatment plants. So I'm showing only one here, but you can imagine if the infections in that area increase, all of these single black data points would increase, but they would not increase independently. The ones in the smaller red and blue circles, these are data from 2 different labs. Because the methods are different, you would expect that these 2 clusters would move together. So there's some kind of dependence in the data in that level of clusters. Within that cluster, there are 2 different things, N1 and N2. We would assume they are also going to vary together. So when these things change from week to week, you can imagine the bubble, each bubble is moving independently, and then the 2 tiny drops within each bubble are the replicate samples that we process. So the variation between those 2 is the only thing that's random or independent from each other. Everything else has a dependence, so they move together. And because we have this kind of intricate data structure, we have been able to use this multi-level regression model, where so all the variables that have some dependence on them, we are treating them as fixed variables, and then the replicates that change randomly, we are assigning them as random variables. And when we use this structure in the data to fit the linear regression model, we can predict whether a particular wastewater sample from 1 week is either going up or going down. And so we are plotting that in the right side here. So the red is showing that it's going up, and if it's not colored, then it's not statistically significant. So either there's like too high of a error margin on that measurement, and if it's going down, which is a positive sign, we are coloring it in green here. And this is showing the trend in a log scale. So 1 here means 1 log of change between last week and this week. And this is the short-term trends that we are doing. So all these samples that appear red here, they were actively being used for targeted interventions by the health department. For the long-term trend, so we have taken this data over approximately 6 months. So for the first couple of months, so starting from May, we did only 16 treatment plants because we were trying to get our method set up. And then from June onwards, we started doing all 39 treatment plants in Houston. All of the data is combined here. So again, same logic as the last week. So if there's data from multiple sources, you want to combine them before you do other statistical analysis on them. So here the data was combined. And so what I'm plotting here is a non-parametric spline regression. So the data from a single treatment plant is appearing as this plus here, and the size of the top arrow in the plus, that is showing the error bar, the error margin for each measurement. So some treatment plants are smaller, so it's obvious that they would have a lower total amount of virus. So what we do here is we take the viral measurement that we do, copies per liter, multiply it with the flow rate. And so we are getting copies per day. And so the smaller treatment plants are lower here, the larger ones are higher. And when we add up all of these, so these data is in a log scale. So the 11, 12, 13 is 10 to the 11, 10 to the 12, 10 to the 13, that way. And so what we have to do is when we have to get the value for the whole of Houston, we have to add up these numbers in the absolute copies and not in the log scale. So you anti-log transform these, so go back into the actual numbers, and then you add them up to get this curve on the top. And this is able to capture very clearly that there was a huge spike around the months of June and July that everyone is aware about. But there's also that before we entered around the time of May, there was also a spike in the wastewater signal here. Just a quick explanation for some people who are unable to understand what a spline means. Very simply put, if you have a bunch of data points and you're unable to fit a single curve to it. So for example, this cannot fit well with a single straight line, or even if you put a curve that's curving down like that. So one way you can fit any curve to this data is to divide it into different intervals. We call them as bins. And each bin is fit with a different curve. So you could fit it with a straight line here, for example, or from what we are doing in our case, each of these is a curve. When the data is a lot more complex, you can fit each bin with a different curve. And when you piece them all together, it appears like there's one single curve passing through, but each tiny fragment of it is given by one function. And so this method called spline is used very extensively when you want to smooth your data. If you have a lot of error margins and you're trying to make observations from your data, it's very useful to smooth your data if you use curved functions to fit it, and also used for predicting data. Use the data to do a prediction for the future. So what we've done is we've taken this green curve here. More or less, it's similar data plotted here, but this is the data of wastewater are aggregated in a different manner. So the log plots shown here are directly averaged to get this. So that's where the axis here starts from around 13. Okay, so I'll wrap up quick. So we were able to find very good correlation, 0.8, between the smooth data from the wastewater and the clinical positivity rate. And we have taken this same data. This was across the whole city. This is within a treatment plant. And you can imagine that within a smaller area, the health data is very noisy, but we're still able to fit it very well with good correlations of 0.8 with the raw, and smooth as even more correlation. And we do it for many treatment plants here. The results seem similar. We also created a model where we can do this spline regression to actually predict. So if you don't give the model the rate, the clinical positivity rate, we can use the wastewater data not shown in this graph to predict the positivity rate. And when we plot them back to back, we see a correlation of 0.8 again. So wastewater actually has information enough to predict the amount of clinical positivity rate, and it's a leading indicator by 1 or 2 weeks from what we have seen in our data. And so in summary, we have compared many methods, identified the best method, and we have used it for long and short-term predictions, and we have enabled many targeted interventions by the health department, which would have prevented many more outbreaks. And we are taking this wastewater epidemiology further to sample 60 schools, 15 nursing homes, and 100 more zip code specific manhole samples. And this is our team that has performed this effort. So we have the health department extensively working with us, and the Houston Public Works providing us with the wastewater samples every week. All the members at Rice University and the statistics team at Rice and the lab at Baylor who were doing this as well. Thank you.
Great presentation, Prashant. So we'll have just about 5 minutes for questions to stay on time. And just as a reminder for all the attendees, if you have a question, please add it in this Q&A box that's in the sort of lower right-hand part of the Zoom window. And also there's an upvote system where you can upvote other people's questions, because that's how we'll choose what to ask with the time we had. So first, before I get to the questions, Prashant, there was a comment that you had a very nice explanation of your methods in the chat. So I think we all appreciated that. So the top question here is from Rose Cantor, and that was, how did you quantify your bovine coronavirus independently to know how much was spiked?
So we used a different primer set for the bovine coronavirus. And so while we were sampling for the N1 and N2, we've simultaneously done a different reaction. Because the abundance of the bovine coronavirus is much higher, we had to dilute the samples before we did the droplet digital PCR. And so because we used a different primer set for each target, we are able to quantify all of them.
Yeah, and I think the question then is, did you do a direct extraction on your spike-in material to understand the spike-in concentration?
Oh, yeah.
Or how did you control for that?
So we did 2 different methods for quantifying the amount of spike-in that we were doing. So one is that we, without putting it into any concentration method, we directly extracted that sample using the RNA extraction kit. The other thing we did is what the data we are showing here is, we directly took the sample and heated it for 10 minutes at 95 degrees. And we see that that releases much more RNA particles than the RNA extraction method. So we were using the boiled virus sample to quantify the denominator for these quantifications. Great,
thank you. And the next question is from Abhi Lasha Shrestha. For the HA filtration method, what pore size filters did you use? For the other filtration method, I believe they're referring to the ultra filtration method, what type of filters were used?
So for the HA filtration, we used a 0.45 pore size filter. And I don't know how much the pore size affects, but we did not try any other different sizes. For the ultrafiltration based method, I think this is a 10 kilodalton cutoff filter. I don't know exactly what pore size it translates to, but I imagine it should be smaller than 0.45 micron.
Great. And what brand did you use, if you know?
It's from Amicon.
Amicon? 10 kilodalton.
Yeah, the ultrafiltration is from Amicon.
Great. And maybe just one last question in the interest of time and keeping us on schedule. So this is from Colleen Naughton. Does the Houston Health Department report the wastewater data on a public dashboard?
We are planning to create one soon. I think it's in the works, but it has not been out yet.
Great. All right. Well, we'll keep an eye out for that. And thank you for the great presentation, Prashant.
You're welcome.
Okay, so Katerina is going to be, excuse me, our next speaker. she can go ahead and share her slides.
Interesting.
Looks
Okay.
good.
Can
Great.
you see this okay?
Yes, it looks good.
Okay, so good morning, everyone. Thank you for the invitation to speak here and share with you some of the results from a few of the projects that we have been working on. My talk is about cells, drugs, and of course, COVID-19, evaluating community health through urban water systems. So the first part of this talk will focus on our NSF CRISP project, the interdependent water infrastructure in our potable reuse system, also known as the unsheltered homelessness. So it's switching a little bit away from COVID-19, but we will of course come back to it in the second part of the talk, which will be about the wastewater surveillance of SARS CoV2 here in Southern Nevada. Well, welcome to the fabulous city of Las Vegas, of course, with its shiny and beautiful, luxurious casinos, restaurants, and all sorts of fancy places. And you know, this picture is probably what comes to mind of most people when they hear the word Las Vegas. It's just rich, rich city, right? However, let's come back to Vegas with, you know, from a different angle, from a different perspective, the one of homelessness. So as I said, there are not many people that, you know, that think and realize that homelessness is actually a considerable issue here. There are more than 6,500 homeless people in the city, and close to 70% of them are unsheltered on a daily basis. In fact, we unfortunately have the 8th highest homeless population in the country and the 2nd highest unsheltered rate in the nation. Homelessness so causes serious concerns. On one hand, you have the socio-economic impacts like poverty, safety, health, and unemployment. And on the other, there is a large environmental impact, notably water pollution and contamination. So as you can see from these pictures, many homeless people tend to live close to the flood control channels that run through the city, obviously to have access to some sort of water. But inevitably, all these channels became filled with trash and waste. And much of this waste also includes human fecal waste. And this is a problem because the contaminated water from the control channels that you can see here on the little map, all that water eventually converges to Lake Mead. And Lake Mead is used not only for recreational activities, but it is also a drinking water source for the city. And so minimizing contamination in the incoming water from the control channels is actually really important downstream. And unsurprisingly, like everyone else, homeless people here in Vegas have been affected by the COVID-19 pandemic as well. At some point early in the pandemic, the shelters were closed because there was a person that tested positive. And so the officials came up with this solution, sort of, to expand into a convention parking lot. And so they painted these lines on that parking lot to demarcate spaces for the homeless people to sleep in. And of course, they had to be 6 feet apart. And this place then became an isolation location, so a quarantine location, once the shelters reopened. And at this point, we are not sampling homeless shelters for COVID-19 monitoring, but I think we are moving in this direction quite soon. And so as you can see, the homeless problem in Las Vegas is not an easy one to solve, but we are hoping to help move things into the right direction. And so to start with, we want to generate data and models to raise awareness of the public health implications of homelessness. And more specifically, we want to evaluate the impact of homelessness on water quality and public health. And ultimately, our goal is to communicate this knowledge and advocate for more effective policy measures to reduce homelessness in the city. So we have been sampling, collecting water samples across Las Vegas, and we have 20 different sites that we collect the water from. We have several high density homeless encampments sites that are close to the Flamingo and Tropicana washes. There are a couple of control sites that are further from the strip and are supposed to be less impacted by homeless population. We have a few confluence sites that receive water from these channels, and of course we sample the wastewater treatment plants. So only 4 wastewater treatment plants for the project, and what we collect is the finished effluent. There are also multiple sites along the Las Vegas wash, so starting upstream with the ULVW site and going all the way down to the Las Vegas Bay, which basically goes straight into Lake Mead. And then finally, there are also 2 sites, 2 locations in Lake Mead that we are sampling at. So more specifically, we sort of collect 2 types of samples. For one set of analyses, the illicit drugs and pharmaceuticals, we collect 1 liter samples just at 8 different sites, which include the 4 treatment plants and then 4 other sites. And for the remaining, so all 20 sites, we collect 2 10-liter carboys, and those are used for microbial and general water quality parameter analyses. So for the microbial analyses, which is the main focus of this talk, each sample goes through a 2-step sample concentration. So in the first step, we take the 10-liter sample and concentrate it to less than a liter. So usually anywhere between 300 to 800 milliliter. And that first concentration step is done through hollow fiber ultrafiltration, HFUF. And then from this concentrate, we take 50 milliliters only and further reduce it to usually less than 1 milliliter with the Centricon filters. So here you can see sort of what it looks in real time. The hollow fiber ultrafiltration is done with a 25S Rexseed dialysis filter. You can see them in the picture on the left, in the left corner. And pretty much the concentration consists in recirculating the sample through that filter while discarding the permeate until the desired volume is achieved. And then the Centricon concentration is done with the 30 kDa Centricon filters. And as I mentioned earlier, we are only using 50 milliliters of the concentrate, so no matter what the concentrate volume is. And this is just for the sake of time and being able to do this project in a timely manner. Or I guess the lab work in a timely manner. So once the concentration is done, so using the Centricon concentrate, we extract the nucleic acid with the PureLink viral RNA DNA minikit, following pretty much the manufacturer's instructions. And then we run a battery of qPCR assays, starting with the 16S rRNA gene to quantify bacteria at all of the sites. And then we are focusing mostly on fecal indicators, so bacteria such as Bacteroides, Enterococcus. Then we also have a few assays targeting the human-specific Bacteroides or canine-specific Bacteroides. And of course, we are also looking at several viruses, including crassphage, which is a phage that infects the Bacteroides bacteria. Then there's a series of human-specific viruses, as well as a plant virus, specifically the pepper plant virus called pepper model virus, or PMMOV. And again, all of these are indicators of fecal contamination. For the illicit drugs and pharmaceutical analyses, we are targeting roughly 50 compounds that can be broadly categorized into analgesics, artificial sweeteners, stimulants, antibiotics, and opioids. Here on the left-hand side, you can see the complete list, but I'm only going to briefly describe findings from 3 of them: the benzoyl agonine, which is a cocaine metabolite, and then we are going to look at caffeine and sucralose. So measuring trace organics can actually help us to better understand the impacts of different factors on water quality. So for example, sucralose can be used as an indicator of wastewater contamination. It is essentially not degraded through treatment, and so we would expect it to be higher or more abundant at the sites collected from the wastewater treatment plants compared to the other sites. And it was actually the case. You can see that the FAT, which is the high density encampment site, and DBC, which is a confluence site, they had a much lower concentration of sucralose compared to the treatment plants. The reason for the 2 other sites, the CON and LVWP, having higher concentrations of sucralose is because they receive discharge from one of the treatment plants. And so this data actually makes perfect sense. We are able to pick up sucralose, which indicates fecal contamination or wastewater contamination, rather wastewater contamination from the different sites if there is contamination or not. Okay, moving on to caffeine. So caffeine can be used to measure the impact of pretty much direct human pollution. And so we see it more abundant in the high density homeless encampment site, the FAT site, compared to the control sites and compared to the treatment plants. Again, it makes sense because caffeine, as I said, it's a direct human pollution indicator. And so it is more abundant in the site that is directly receiving pollution from humans, so the homeless people. And you can see that it was actually completely removed from one of the treatment plants. And last, we also wanted to look at the impacts of homelessness, directly homelessness, on water quality by measuring the level of several drugs. So here I am showing the cocaine metabolite benzoyl agonine, which we do pick it up at the homeless site, but not at the control site. Again, so the confluence from the control sites, and that makes sense because that site is not pretty much impacted by the homeless population. And we do measure it at the treatment plants because this compound is actually relatively stable and not removed by treatment. And because it is stable, we also picked it up at the 2 sites, the CON and LVWP, because as I mentioned previously, these 2 sites do receive discharge from the treatment plants. Regarding bacteria, you can clearly see that they were quite abundant at all of the locations. The control sites, which are shown in green, perhaps you can see that they had somewhat lower concentrations compared to, for instance, the high density encampment sites, and so did the confluence, the DBC, the confluence of the 2 control sites. It also showed that it had lower concentrations of bacteria. Otherwise, there are not really any major differences at the locations along the Las Vegas Wash, which are shown in purple. But for the treatment plants, you can see that the first one had somewhat of a lower bacterial load compared to the rest. And then we do obviously quantify bacteria at Lake Mead. So I should probably mention that this is an average of the collections that we have had so far. So we had 3 dry event collections, and this is the average concentrations across the 3 time points. There were not really any major differences, major temporal differences. And so for the sake of simplicity, I just decided to combine all of the data and show it at once. Regarding the fecal indicators, so here you are looking at the concentrations of 3 targets: the Bacteroides bacteria, a crassphage, which is the Bacteroides phage, and then PMMOV. As I already mentioned, it is a plant virus, and it gets into wastewater through human consumption of infected peppers or pepper products. And clearly, the fecal indicators were detected at the sites close to the homeless encampments, because it is not uncommon for homeless people to straight up pee and poop into the flood control channels. the pepper mild model virus was most abundant at the 2 treatment plants that use UV disinfection, and it is because PMMOV is actually resistant to UV. On the other hand, it is sensitive to chlorine disinfection, which is used by the 2 other treatment plants. And so you can see that it was barely detected at these 2 sites. And then lastly, we did pick up PMMOV along the Las Vegas wash in the sites that did receive discharge from the treatment plants. And then last but not least, a good thing was that we did not detect SARS-CoV-2 at any of these sites. So we ran N1, N2 at all of them, for all of these sites, and did not detect it even at the treatment plants. And that is because we are collecting the finished effluent. And so this is a good indication that the virus is degraded through treatment and we are not picking it up at the end. So in the finished effluent. So speaking of SARS-CoV-2, we have been monitoring for this virus in our local treatment plants since March 1st. We started at these 2 facilities, which are the major facilities in Las Vegas, and then a couple months back, we moved and expanded to all 5 treatment plants. So currently, we are covering the entire city of Las Vegas and monitoring the virus across the whole city. For the first 2 facilities, we use the HFUF, so similar to the NSF project, and we are processing 10-liter samples through the same Rexheat filters. For the remaining facilities, we are using the Centricon filters and only working with a smaller sample, which is 150 milliliters. All of our samples are spiked with the bovine coronavirus that we use as a sort of process control and then to calculate our viral recovery. Again, similarly to the NSF project, we are extracting RNA and DNA with the PureLink viral RNA and DNA minikit, and then make cDNA with the Maxima first strand cDNA synthesis kit. And the cDNA is then analyzed for 4 different targets of SARS-CoV-2. So obviously the CDC, N1, N2 nucleic acid proteins, but we are also targeting the envelope protein with the ESAR-BACO assay, and then the open reading frame 1 with the nCoV assay. We also quantify PMMOV as a fecal indicator in a sort of a process control, and then obviously our spike, the bovine coronavirus, to calculate virus recovery. So at facility 1, we have been monitoring for SARS-CoV-2 since March 1st, but the signal did not start showing up until about March 9 or so. However, unfortunately, since then, we have been consistently detecting it, and you can see that the concentrations relatively nicely match the number of new cases. So the number of new cases are shown in the blue bars, and on the x-axis, you can see that we are going from March all the way to November. The SARS-CoV-2 concentration is shown in the green symbols, and it is an average corrected for bovine coronavirus recovery, and it is an average of the 4 different targets. And in red is the wastewater model, which is based on the number of new cases each day and the shedding rate. And the match between the model and the actual SARS-CoV-2 wastewater concentration is not perfect, as you can see, but I think it is also not expected to be, because there are still many uncertainties regarding, for example, the shedding rate, the actual daily reported cases, and so on. Both of which are used in calculating the predicted concentrations. Nevertheless, I think that the wastewater concentrations do follow overall a very nice and close pattern to the one you can see from the cases. And that is especially the case for the last 2 months or so, where the cases have been on a rise, and so has the wastewater concentration of SARS-CoV-2. And this is essentially the same figure, but for facility 2, which is a substantially smaller one and serves a smaller population. We only started sampling this facility in May, And again, there is a lot of variation, perhaps more than for the first one. But again, the overall pattern between the concentration of SARS-CoV-2 in wastewater and the model is pretty close. And again, you can see a decent match for the last few weeks. And last, here you can see the concentrations in log scale of the SARS-CoV-2 gene copies per liter of wastewater at all of the facilities that we have started sampling in August. So 1 and 2 are the same that I just showed you, and then 3, 4, and 5 are the 3 other ones that we started sampling later. And for the most part, if you look at each given facility, for the most part, the concentrations were roughly constant until the month of November, where every single one of them is on the rise. So whether it was a 10 to the 4th or so copies per liter and increasing to 10 to the 5th, or 10 to the 5th and increasing to 10 to the 6th. The facility 5B is actually, I think, the most important, most interesting one, because the first time we sampled it, we got the highest ever concentration, up to 10 to the 7th gene copies per liter. And then the following week, it dropped down to essentially the lowest concentration that we have seen. And since then, it's been fluctuating much more compared to the other ones. Regarding our future plans, so both of the projects are ongoing. We still have 4 more sampling events for our NSF CRIS project, which one of them should be a dry event, and that is not a problem, but 3 of them should be wet events, which is a problem because we are not sure if it's ever going to rain again in Vegas. And so we'll see if we are able to pull this off or not in a relatively short amount of time. And of course, we are going to continue our monitoring efforts for coronavirus across Las Vegas. And as I mentioned, we are probably going to be expanding to sampling the homeless shelters, nursing homes, and there was also word of possibly sampling the university. But again, we are not sure about this yet. And with that, I want to thank Dan Garrity, because simply without him, none of this would be possible. Essentially, it's just him and me doing all of this work. And so really big thank you to you, Dan. And then I also want to thank the staff at our collaborating wastewater agencies, staff here at SNWA, all of you researchers who are participating in this COVID-19 pandemic efforts. It's not an easy task, mentally and physically, it's exhausting, but I feel like it's very important that we do this. And of course, I also want to thank UNLV and the Genomics Core Facility and the Department of Civil and Environmental Engineering and Construction. Okay, thank you for listening, and I'll take questions.
Thanks so much, Katerina. That was a great talk for a kind of population that doesn't get a lot of attention, doesn't have a lot of resources. So I'd like to applaud you for taking this on. Clearly, it's very challenging work. But it is difficult to do, and resources are always rare. We have a few questions here, but maybe I'd like to start out. Homelessness is very difficult to study, even to count people. One of the questions here was, does the city provide kind of porta potties, which would have been an obviously good location to take samples? Did they provide that, and did you make any attempts to possibly use infrastructure provided by the city for the homeless to then sample those particularly?
No, not yet. So as far as I know, there are not really porta potties available for homeless people. There are shelters, but my understanding is that they actually do not necessarily like to be in shelters. So even if the shelters are not completely filled, homeless people do not necessarily choose to go in and stay in, especially not right now in the pandemic. I'm not sure about the sampling yet. So there has been word of us expanding into the homeless shelters, but I don't have details on it yet.
Yeah. Now that would require internal review board, right? If it's a sensitive population.
Yes, yeah. It is not as simple as going to the treatment plants.
Yeah, we have another question from Colleen Norton, who is also, as I am, very interested in your data, and we're wondering how you're sharing that information. Is there a public dashboard? How is this data shared that you collected, not for the homeless now, but for the city of Las Vegas?
so the Southern Nevada Health District has a dashboard that provides all of the daily cases. We unfortunately do not have this data publicly available yet. So we are just providing it back to the treatment plants essentially for their own interest. But I feel like that is a major missing part that we need to attack, is to provide this data and make it publicly available so that it has some sort of a direct impact or helps the city to make better decisions. But we are not there yet.
Thank you. Yeah, it is regrettable that that is a privilege to have access to data, and the data are not shared quickly and also not completely with the people who actually are affected by the data. So I personally, I don't know if we have people listening in who are aware of additional public dashboards that display COVID-19 or SARS CoV2 data for sewage. I encourage you to add that to the chat box. Obviously, we do this in Tempe, Arizona. Since March, we have the first dashboard up, and I know there's a few others, but if we have more, please add them. And I think maybe we can use that as a community to kind of motivate our city leaders to get those data out. I'm encouraged to hear that in the previous talk by Prashant that we have efforts there to do this in Houston. Good. We have, let's see, and we have another question. We have a paper here. No, I think we are exhausting our questions.
Well, it's time to wrap up anyway. So thank you everyone for attending, and thank you to our speakers who did a great job. And we do have another webinar before the holidays, which I will and my colleagues will be sending you notes about on December 2nd. So we hope to see you all there. Have a great day. Goodbye.
Bye. Thank you.